Okay, thanks to Franklin and Watson and Crick and everybody that we talked about in chapter 16, we know that DNA is the molecule of inheritance. Next question is, how? Right? How do we express DNA? How does a gene code for a characteristic, a trait, a structure? Well, there was a person by the name of Archibald Garrod who first suggested that maybe genes dictate phenotypes by coding for enzymes. We know that enzymes catalyze chemical reactions. It seems logical that if your DNA codes for your enzymes, right, then they dictate the kinds of chemical reactions that go on in your cell, and they dictate what happens in your body. Well, he didn't have any actual evidence for that, and so later on, George Beadle and Edward Tatum did some very, very famous experiments by causing mutations on bread mold, by exposing them to X-rays, and creating mutants and seeing what kinds of media they could grow on. And by doing that, they got an idea of the types of enzymes that they had. So they knew they were mutating DNA. They were seeing if the DNA impacted their ability to metabolize products, i.e. did DNA affect which enzymes they had. So the wild-type bread mold, right, the bread mold that comes from the wild, that doesn't have any mutations from these X-rays, is able to grow on what we call a minimal medium. Right, so this is only the stuff that it needs to survive. But once you start causing mutations, these mutants aren't able to grow on that minimal medium. Now, all of them can grow if you add something called arginine. Right, arginine allows all of the mutants, in addition to the wild type, to survive. But arginine is something that you can create, typically, if you are a bread mold, inside your own cells. So they hypothesized that somewhere in the pathway seen below to produce arginine, these mutants had a messed up enzyme, right? The X-rays, which messed up their DNA, messed up their ability to make one of those intermediates, right? One of those enzymes that turns this precursor into ornithine, or ornithine into citrulline, or citrulline into arginine, right? You need all of these enzymes in order to cause the precursor, which is found in the minimal medium, to turn into arginine. So they figured out that with these three different class mutants, they had knocked out different enzymes, right? So class 1 mutants couldn't survive on the minimal medium, but if you gave them arginine, they could survive. If you gave them citrulline, they could survive. Even if you gave them just ornithine, they could survive because they still had the enzymes to turn the ornithine to citrulline to arginine, right? Class 2 mutants, they couldn't survive even with ornithine because it was later down the line that their enzyme was knocked out, right? You had to give them citrulline or arginine, right? In class 3 mutants, it was this last enzyme. So you could give them ornithine or citrulline. Either way, they couldn't turn this last bit into arginine, so you had to give them arginine to survive. This being some actual empirical evidence, right, that the DNA, which they had messed up with these X-rays, is coding for enzymes, right? And so they sort of proved Archibald correct, or at least supported his hypothesis. This eventually led to widespread support of the one gene, one enzyme hypothesis. So genes are bits of DNA that code for enzymes. Now, we've since expanded that a bit because genes code for proteins, and not all proteins are enzymes, right? Some of them are structural, some of them are pigments, some of them are channels that allow certain things across membranes, right? We've talked about all the different things proteins can be, and we've expanded this to a one gene, one protein hypothesis. So our definition of a gene, then, is a stretch of DNA, a chromosome, stretch of A's and T's and C's and G's, that code for a particular protein. Now, it's actually more complicated than that, but we'll get into that later. For now, a gene codes for a protein. So now that we know that the way that genes code for traits, right, is by coding for proteins, right, well, how do genes code for proteins? Well, there are two main steps in this, transcription and translation. And transcription is transcribing, right? Like you transcribe, right, a phone call, or if you're dictating a letter for somebody, right, you make a note of what they say, a copy, right? And so transcription is the process of making an mRNA, messenger RNA copy of what the DNA says, of the information in the DNA. And that's important because DNA can't leave the nucleus, and the information has to leave the nucleus to get to where we're building the proteins, which are the ribosomes. So mRNA is the mechanism by which we do that. So transcription is the production of this messenger RNA. Translation is the using of this mRNA to create a protein or a polypeptide. So DNA and RNA are both in the language of nucleic acids, right? They're nucleotides that are put together. Proteins or polypeptides are in the language of amino acids. They are amino acids put together. So you essentially need to translate from the language of nucleotides into the language of amino acids. And that's why this using of the information from mRNA to make a protein is called translation. So the two main parts are transcription and translation. Transcription occurs in the nucleus. Translation occurs at the ribosomes. Now, if you're in a bacteria or some sort of prokaryote, translation of the mRNA that you're producing during transcription can begin before you're even finished making the mRNA, because there is no nucleus, right? So the ribosomes are close to the DNA, and so as you're making an mRNA, it can bind to a ribosome before it's even finished, right? That cannot happen in a eukaryotic cell because the nuclear envelope separates the area where you transcribe with the area where you build the proteins. So eukaryotic transcription and translation are going to be similar, but they're going to have some key differences. So the informational flow in a cell is thus: DNA in your nucleus codes for RNA, which is able to leave the nucleus, and that RNA then codes for a protein at the ribosome. DNA to RNA to protein. That's sometimes called the central dogma of biology, right? That is the direction of informational flow. But like everything, there are actually some exceptions, and we'll talk about those when we get to viruses. But DNA in the nucleus, right? Or not in the nucleus if you're a prokaryotic cell, right? But DNA codes for RNA, RNA codes for a polypeptide or a protein. As you may recall from when we talked about macromolecules, the monomers which make up polypeptides or proteins are called amino acids, and there are 20 of them. And that's a good number of letters in the alphabet, so to speak, right? You can get an incredible diversity of structures because you can get an incredible diversity of sequences of amino acids. This is also why a lot of people thought proteins must have been the language of inheritance, right? The information carrying molecule, before we figured out that no, it's actually DNA. But you can get a lot of information into a short alphabet, right? Even a 4-letter alphabet like DNA. For instance, Hawaiian. I spent a semester in Hawaii in college, and the Hawaiian language is really interesting because it has many fewer letters than the English language, which has 26. And you can still get as many words in, they just have to be longer, right? So the state fish of Hawaii is called the Humuhumunukunukuapua'a. It is the Humuhumunukunukuapua'a. I may have gotten that right. Anyway, words get longer to carry the same amount of information. Even in a 2-letter alphabet, right, you can have really long words and carry the same information, essentially, right? So think binary code in a computer, right? If you put enough zeros and ones together, you can make this happen, right? You are seeing me and hearing me because this computer was able to turn all of this stuff into zeros and ones and send it to you across a series of tubes, and then you were able to upload that series of zeros and ones and turn it back into my face and my voice, right? So 4 letters is sufficient. But to code for each amino acid, how many nucleotides does it take? Well, if it was only 1 nucleotide per each amino acid, you could only code for 4 amino acids because 1 nucleotide, adenine, say, would code for 1 amino acid, thymine would code for another, guanine would code for another, and cytosine would code for another, and that's 4 amino acids. So we need to make our words longer, right? They can't just be 1 nucleotide long, they have to be at least 2. Well, with 2, you can have 4 options at 1 and 4 options at the next site. That's a total of 16 options. Is that enough? Well, no. We have 20 amino acids. So our nucleotide words can't be 2 letters long. They have to be at least 3 letters long. So we call it the triplet code. Now, 4, 4, 4, right? That actually leads us with 64 possibilities, which is more than we need. That's okay. That gives us some redundancy. So some triplets, you'll have more than one triplet DNA code that will code for the same amino acid. That's good because even if you have, maybe a mutation here or there, sometimes that mutation won't affect the sequence of amino acids that you're building into your protein. So, for instance, your DNA template strand, ACC, right, would code for an mRNA that is complementary UGG, and UGG always codes for tryptophan, right, as an amino acid. So DNA is transcribed into this mRNA codon. We call this a triplet code. We call this on the mRNA a codon, and then this is an amino acid tryptophan. Note a couple of things. First of all, that this UGG, UUU, etc., is not an exact copy of ACC, AAA, right? It's the complement of it, right? Now, if there's a C here, there's going to be a G here, right? So it carries the information, but it's not an exact copy. So the mRNA is not actually a copy of the DNA, but it carries the same information. In the same way that if you guys know anything about photography, before digital photography, you would have negatives, and you could use your negatives to get more of your pictures printed. The negatives were essentially wherever it was light in your image, it would be dark on the negative, and wherever it was dark in the image, it would be light on your negative. So right here, right, where this window is behind me, if you took a picture on the negative, that would be very, very dark, right? My beard would be very, very light in the negative, although getting a little bit of salt in my pepper here. So it contains actually the complement, right, the opposite of what you had, but it still carries the information because from what you have, from that complement, you can go back and know what the original said. So from ACC, you get UGG. The second thing to note is A binds with U, or A codes for U instead of T. If you remember, RNA has the same nucleotides as DNA with one exception. Instead of T, thymine, right, you have U, uracil, right, which works better in a single-stranded molecule. And then UGG, or UGG, codes for tryptophan. Tryptophan, by the way, is what's in turkey, and they say makes you sleepy. I think that's false, but I always remember that because when I fill up with turkey, I go, ugh. If I give you a strand of DNA, a template strand, I would want you to be able to tell me what the complementary strand of DNA was, right, on a test, say. I would also, if I gave you a template strand, want you to be able to tell me what the the messenger RNA strand would look like, right? What codons would follow from these triplet codes here on the template strand. I would also want you to be able to tell me what amino acid sequence you would get. Well, how do you do that? Well, you do it not out of your brain, or I'm not going to make you memorize these, but I would give you something like this, right? This is a chart that shows all of the mRNA codons and which amino acid they code for, right? So these 2 code for phenylalanine, right? These 4 code for leucine, these 4 code for valine. Note that there is redundancy, right? More than one triplet, or mRNA codon, I should say, codes for the same amino acid. So if you get a very small mutation, sometimes that small mutation will have no effect on the actual structure of the protein. So again, I want you to be able to take a DNA template strand, give me the complementary strand of DNA, or be able to give me the strand of messenger RNA that it would code for, and ultimately be able to give me, if I hand you this chart, the sequence of amino acids that you would see. Note that AUG is a start codon, and that these 3 are all stop codons. That will be important when we get to translation.